contents for this video is introduction, structural isomerism, geometrical isomerism, optical isomerism, and generality, and the last one is real-world application and conclusion. Objective. Our first objective is to define and differentiate between structural and stereoisomerism. Second objective is to explain and illustrate geometrical isomerism in coordination number 4 and 6. The last objective are to identify and explain optical isomerism in octahedral complexes. What is coordination compound? Compound that typically contain a complex ion or counter ion, while isomerism definition are same molecular formula but different arrangement of ligands. There is two major categories. The first one is structural isomerism. Structural isomerism are compounds that have the same molecular formula but different atom-to-atom connectivity. The second one is stereoisomerism. Stereoisomerism are compounds that have the same molecular formula and the same connectivity of atoms but different in the 3D spatial orientation of the atom.
What is structural isomerism? Structural isomerism refers to the coordination compounds that have the same molecular formula but different connectivity or placement of ligands. In other words, the components are the same, but they are arranged differently within the compound. This difference usually depends on what is inside the coordination sphere and what is outside. Structural isomerism in coordination compounds can be divided into four main types. These are hydrate isomerism, ionization isomerism, linkage isomerism, and coordination isomerism. I will now explain each type one by one using simple examples. Hydrate isomerism occurs when coordination compounds differ in the number of water molecules inside the coordination sphere. In one compound, all water molecules are directly bonded to the metal ion. In another compound, some water molecules are outside the coordination sphere as water of crystallization. Even though both compounds contain the same components, the position of the water molecules is different, which result in hydrate isomerism. Ionization isomerism occurs due to an exchange between a coordinated ion and a counter ion. This means that ion that is originally bonded to the metal may move outside the coordination sphere, while a counter ion moves inside. As a result, the structure of the coordination compound changes even though the molecular formula remains the same. Because of this exchange, the compound releases different ions when dissolved in water. This leads to different chemical behavior in solution, such as forming different precipitates. So, ionization isomerism affects which ion is released into the solution. Linkage isomerism occurs in coordination compound where the donor atom of the ligand is different. This type of isomerism involves ligands that can bond to the metal ion through more than one atom. In linkage isomerism, the ligand remains the same, but it attached to the metal ion through different donor atoms. For example, a ligand may bind through nitrogen in one compound and through oxygen in another. Although the ligand is the same, the point of attachment changes, producing different linkage isomers. Coordination isomerism occurs when there is switching between metal atoms in two complex ions. In this case, both the cation and anion are complex ions. The ligands are reorganized between two metal centers. More specifically, coordination isomerism involves the exchange of ligands between complex ions. The overall molecular formula remains the same, but the coordination environment around the metal ion changes. This results in different coordination isomers. So far, we have seen that structural isomerism changes how ligands and ions are connected or positioned in coordination compounds. Next, we will move on to the geometrical isomerism, which focuses on the spatial arrangement of ligands around the metal ion.
Next, in this session, I will talk about geometrical isomerism, which is one type of stereoisomerism in coordination compound. Geometrical isomerism occurs when coordination compounds have the same molecular formula and the same bonding, but different in spatial arrangement of ligands around the central metal ion. This type of isomerism is clarified as stereoisomerism. Other than that, it's also mainly occur in square planar and octahedral complexes because this geometrical allow ligands to occupy different relative position. Next, we move on to the square planar cis and trans. For square planar complexes, geometrical isomerism occur as a cis-trans isomerism. Using PT-NH32 Cl2 as an example, in the cis isomer, the two chloride ligands are positioned next to each other, while in the trans isomer, the chloride ligands are opposite to each other. Next, we move on to the importance of cis-trans in biological activity. Although both isomers have same formula, but geometrical causes different biological activity. For example, cisplatin is effective as an anti-cancer drug, while transplatin is not. Next is octahedral Fesmer. In the octahedral complexes, geometrical isomerism occur as Fesmer isomerism. For a complex with the formula Ma3B3, the Fec isomer has three identical ligands on one face of the octahedral, while the Mer isomer has the ligands arrangement around a meridian.
Now I'll start with optical isomerism. Optical isomerism occurs when a molecule exists as non-superimposable mirror image. These isomers have the same formula and bonding, but differ in 3D arrangement. Importantly, no bond breaking is needed to convert between them. These isomers are called enantiomers, just like our left and right hand, which are minor image but cannot perfectly overlap. For optical isomerism to exist in coordination complexes, three main conditions must be met. First, the complex must have an octahedral geometry. Second, it must contain bidentate ligands such as ethylene-diamine. And third, the complex must be asymmetric, meaning its absence of plane of symmetry. Without all these three conditions, mirror image isomers cannot form.
Next, let's look at a common example. This is an octahedral cobalt 3 complex. It has two bidentate ethylene diamine ligands and two chloride ligands in the cis position. This complex exists as two optical isomers. First, delta form, right-handed twist. Second, lambda form, left-handed twist. Delta and lambda are non-superimposable mirrors image, just like left and right hands. Finally, why this matters in biology? First, biological systems are chiral. Second, proteins and enzymes exist in specific stereochemical forms. Third, enzymes are stereospecific, meaning they only interact with one enantiomer. And fourth, chirality is critical for biological functions, such as oxygen binding in hemoglobin.
Let's look at a real-world example that shows why isomerism truly matters. Cisplatin and transplatin. Both compounds have the same molecular formula and the same metal ion, platinum. However, they differ in geometry. In cisplatin, the two chloride ligands are positioned next to each other, while in transplatin, they are placed opposite each other. This difference may seem small, but its effect is huge. Only cisplatin can bind effectively to DNA. Its geometry allows it to form cross-link between DNA bases, which prevents DNA replication and stop cancer cells from dividing. On the other hand, transplatin cannot bind DNA in the same way, making it biologically inactive. This clearly shows that even when the formula is the same, geometry determines function. Another powerful example of coordination chemistry in real life is hemoglobin. In hemoglobin, the central metal ion is iron 2. This iron is coordinated to nitrogen donor atoms within a porphyrin ring. The coordination geometry around the ion is extremely important. It allows hemoglobin to bind oxygen molecules reversibly, picking up oxygen in the lungs and releasing it in body tissues. If the geometry around the iron were altered, oxygen binding would become inefficient or even impossible. This shows why stereochemistry is critical in biological system. The correct three-dimensional arrangement ensure proper biological function and ultimately support life itself. To conclude, isomerism in coordination compound can be divided into structural isomerism and stereoisomerism. Structural isomerism involve differences in how ligands are connected, while stereoisomerism involve differences in the three-dimensional arrangement of those ligands. As we seen through examples like cisplatin and hemoglobin, even small changes in structure can lead to major differences in function. So the key message to remember is this: in coordination chemistry, structure controls function.